Good morning, student. Myself is Dr. Shivaji Andhale, Department of Botany, S.V.K.T. Arts, Science and Commerce College, Deolali Camp. On behalf of Maratha Vidya Prasad Samaj, Nasik. Dear student, today we will discuss about, in previous lecture, what is a plant pathology. What is a pathology? Means phytopathology. Phyto means plant. Pathos means suffering. And logos means discourse or you can study to them. These pathology they are the Greek word and a detailed study to them by these different types of the diseases. Disease cycle. First there is the appearance of the spore on surface of this plant and they start the germination on surface of this epidermal cell and they germinate to them, there is the formation of the mycelium. After they can be grow to them throughout the whole this part of this plant and there is a fully development of this sporangiophore on surface of the body and totally these plants are destroying to them and after they can be developing to them this formation of the sporangiophore. Inside that sporangiophore there is a thousands of the spore they can be developing to them. There is a multiplication or mitosis or meiosis can be takes place inside that this single nucleated structure and there is a large number of this micronucleated spore they can be developing to them in the sporangiophore. After they can be developing of the sporangiophore, there is a very short their life cycle and the rupturing to them at a favorable condition and inside that sporangiophore they release thousands of the spore inside that atmosphere and these spores they can be passed through their one plant to the another plant or same plant of the another part of this plant, one leaves to another leaves by using certain of this agency like wind, insect, human being. Different certain of the agency. There is a very useful or beneficial for the transfer of the disease from one plant to the another plant. Also, there is a irrigated water. There is also very responsible for transmission of this disease from one field to the another field. This is called as a disease cycle. Then the stages in the disease development. There is a different stage. In a pre-stage, mostly human beings they don't know about the appearance of the disease on surface of the plant. Pre-stage is the premature stage. First only the spore are germinated and they can be inserted inside that leaves. And after they can germinate them, they are starting their stage. From one this procedure can be settled down on the surface of the leaves. This is the first stage. And after at the favorable condition, these spores they can be start their germination. They start their development. And certain of the spore there is the entry of the spore into their cuticle. Certain of the spore they can directly enter through their natural opening stomata. Or certain of them can be penetrated by the epidermal cell and they can be developing this spore inside that plant body. These are different stages from this development of these diseases. Then today's lecture we will discuss about then introduction to plant pathology, then host, symptoms, parasite, pathogen, inoculum. First we discuss about the disease cycle. Disease, it is the plant pathology. Disease, this is the science of this plant disease is called as a plant pathology or phytopathology. And this is a phytopathology or this is a plant pathology means phyto means plant. Pathos, pathos means there is a suffer by the any types of disease and logos means logi means logos means there is a study to them. It is called as a plant pathology. Disease cycle, the chain of events that lead to their development of the disease is called as a disease cycle, which may be in a different to their pathogen from the life cycle in a different species. The incidence and severity of the majority of the plant diseases vary on a distinct cyclic basis. The first requirement for the establishment of the disease is the pathogen to come to contact with the host. First the contact on surface of the leaves as well as any part of this plant. And after they can be come to them in the surface of this plant, they can be the certain of the agency, wind as well as the insect or also certain of this animal, the wind they can be carried to them from one plant to another plant. Wind is the most important way in which fungal spore or rust spore or this is a powdery mildew are disseminated over a very long distance. Why they disseminated from the very long distance? Due to these spores are very tiny, very light weighted. They are easily dispersed from one place to the another place. and hence there is a major factor for the spreading of the disease is the wind. Then water is also very important thing for some of the fungal pathogens. Very essential water. For that, it can be transmitted of this in irrigated soil. This is a septoria, or it can be spread, or especially over a short distance from one location to the another location. These are transmitting to them. The set of the stages in the disease development. First is the inoculation. Second thing is the penetration. Third thing is the infection. After fourth thing is the growth and reproduction. And fifth is the dissemination of pathogen. These are the different stages they develop from this on surface of this plant diseases. First, we will discuss how the inoculation, how to inoculate this arrival of this pathogen on the surface of the host. The inoculum, what is my inoculum? The pathogen that land on the host. They can be land, they can be permanently on surface of this any part of this plant, and any part of this pathogen that can be initiate the infection on surface of this plant. During the unfavorable condition like sudden change in temperature, suppose there is a high temperature or low temperature, effect of this poisonous gas inside this plant surface, disturbance of the soil moisture also, immediately there is a decreasing the soil moisture, there is also directly adverse effect. Also there is a relation that pathogen may occur inside the there is a perinating organ or organs. With the insect of this favorable condition, this is with the onset of the favorable condition only. Unfavorable condition, these spores do not germinate to them. But at the favorable condition, these spores there is a germinate to them. It become active and causes the disease. The portion of this pathogen that is responsible for infection is known as a inoculation. Means any types of the pathogen, any types of the spore, they are responsible for infection on the surface of these leaves, and they can be cause a disease. In presence of the susceptible host, virulent pathogen and favorable environmental condition, a disease may not develop due to variation in amount of the inoculum. If the amount of the inoculum is less in the deposit, the pathogen will get more food and will germinate properly. Means the amount of inoculum, there is a available of favorable condition, each and everything they can be available to them of the pathogen or of the spore, food as well as in climatic condition, means moisture is abundant, at that time there is so much favorable, and these spores they can germinate to them. So optimum concentration of the inoculum is essential to develop the disease. The amount of this inoculum which is responsible for successful infection is known as the inoculum potential. Then type of the inoculum, first is the primary inoculum. In a primary inoculum, an inoculum that survive the dormant period and cause the first infection. At the dormant stage, plant they do not grow to them properly, they can be at a specific stage. At that time, these infection they can be occurring to them, for their infection on surface of this plant. Then primary infection caused by primary inoculum, they inserted to them. Then next is secondary infection, caused by the secondary inoculum. Pre-penetration. What is my pre-penetration? Pre-penetration phase, they include the growth of the pathogen. Before actual entry, actual penetration into their host cell, it is called the pre-penetration. Means there is a landing on surface of the leaves. It is a pre-penetration. Spore of the various pathogenic fungi germinate on the surface of the host plant at the favorable condition. Spore germination beside various physical factor, like majority there is a moisture is very essential. Next, another thing is the temperature, pH, light, oxygen, carbon dioxide. This is a major factor for the germination of the spore. In many influenced by non-parasitic microorganism present in the rhizosphere and phyllosphere. During the germination, the spore produces there is a filamentous germ tube and form one or more germ pore. The formation of the germ tube is affected by the environmental factor and is susceptible of the plant. Next thing is the penetration. The first is the penetration. This next thing is the penetration. Spores show a various penetration mechanism. The infection thread of the pathogenic fungi may enter the host through their natural opening. Natural opening is very well known about the stomata, lenticel, hadathode. This is the natural opening of the plant, and majority of this spore of this fungal or bacterial or viral, they enter through this natural opening and they cause the disease. Through this wound or by the direct their penetration. First, it can be happening wound formation on surface of this plant, or directly they enter through this natural opening. Most of the rust or downy mildew enter through this stomata. Most of this are rust plant, or this is microscopic powder mildew as well as downy mildew as a rust. These spores that directly penetrate, they can be directly enter through the natural opening stomata, lenticel and hadathode. A blade-like wedge grow from this appropriation. This is a aprosorium through the stomatal slit, and it swell to form substomatal vesicle. The stomata, stomata is a major factor. Hadathode and these are nectaries are their common pathway for entry of this many parasitic bacteria, many fungi, many viruses for this natural opening. All powdery mildew and few of the downy mildew usually penetrate host tissue through their epidermis and by mechanical process. Landing of the arrival of inoculum. Most is carried by wind, second is the water, insect. These are major, they can be landing to them, they can be passing to them from one plant to the other plant. Only small number of them actually land on the susceptible plant. Very small, very microscopic of the spore, they can be land to them on susceptible surface of this plant. Best example is germination of the spore and seed fungus. Then after they can germinate, the hatching of the nematode on eggs. Nematode, it is an insect, they can be hatching their eggs on the surface of this plant and attach to this pathogen to the host, and after they can be recognition between the host and pathogen, the attachment to them. Then initial invasion of the host by this pathogen, directly they penetrate to them, directly entry to them, penetration through their natural, mostly this is a natural opening. They enter inside that cell and penetrate through their wound and directly their penetration. Then penetration, this is the second thing, direct with their hostoria. There is a formation of the sort of button-like structure. It is called as hostoria inside that cell. Then directly this is a subcuticular only. Then direct this is a intercellular mycelium. Also this is directly that intercellular mycelium with their hostoria and direct with the aprosorium. There is a formation, you can penetrate by the own surface of this plant. There is inside that cell, there is a formation of hostoria, button-like structure, direct substance only, there is mycelium formation and that type. We will discuss in one by one. The most common type of the penetration by fungi and nematode and only type of the parasitic plant. Fungi use fine hyphae produced by the spore or mycelium, penetration peak produced by an aprosorium. This is formed at the point of contact with the host. Penetration is through the mechanical force and is obtaining a cell wall by an enzyme. There is an enzyme, they can be secrete to them. First they can penetrate to them, they insert to them, and after a certain time they can be released certain of the enzymes. Most of the fungi form an aprosorium at the end of the germ tube. Then penetration peg emerges from that flat surface of the aproporium, and there is a precise the cuticle and cell wall. Penetrate through the cell wall also in this the cuticle, they can be also entering to them. Then penetration through their mechanical force, sort of mechanical force can be applied to them, and this softening of the cell wall by an enzyme. Sort of the enzyme, some of fungus, they release certain the enzyme on surface of this plant. Suppose there are spore, they can be deposited on surface of this leaves or stem, and at that time that leaves there is this spore, they can be released certain of these enzymes, and these enzymes they are very useful, very beneficial for entry, for decomposition, for the smoothness of the cell wall, for germination of these fungal spore. Most fungi form an aprosorium at the end of the germ tube. Then the penetration peg emerges from the flat surface of the aprosporium, and there is a passage the cuticle and cell wall. The peg grow into their fine hyphae. This is a very small hyperstructure, thread-like structure, reach in normal diameter, very small diameter. Once it is inside the cell, and as in the case of the apple scab, the fungus penetrate only the cuticle and stay between the cuticle and the cell wall. The parasitic plant also penetrate the same way. This is therefore direct with the Hostoria formation. This is the above surface of this is the blackish spot. There is a they are these spore. They start the germination on surface of this plant, and they enter through their natural opening, and there is formation of this button-like structure inside the cell. It is called as a Hostoria, and it is called as a superficial mycelium. They can be developing on surface of this plant, and each of the natural opening they can be enter through their mycelium, and the formation of the button-like structure, and that Hostoria they can be very useful or very beneficial for the absorption of the nutrient inside that cell, and there is a developing continuously by this above surface of this mycelium thread, and these Hostoria they can develop throughout the whole their cell of this plant of these leaves, and totally these leaves are they stunted their growth, and ultimately the leaves certain of time, certain of duration, there is a elevation color. Then next thing is the directly subcuticular only. When this spore there is a settle down on surface of the leaves, then they can be germinate to them, and they pass inside that cuticle just above the epidermal cell, and they start their division, start their mycelial formation, branching formation inside that cuticle, and certain of this their sphering like structure they can be developing to them by the cuticle and epidermal zone. And that process is called as a directly subcuticular penetration. The next thing is the direct intercellular mycelium. Certain of the spores, certain of this fungal spore, they germinate to them through natural opening, and they pass through them from one cell, and inside there is the formation of their branching like structure or thread like structure of the mycelium inside that cell, and that process is called as the intercellular mycelium. Next thing is the direct or intercellular mycelium with their Hostoria. Certain of the spore they can be germinate to them, they can be penetrate to them, and there is a grow their branching formation. After branching formation, there is formation of their sub-branching, and each sub-branching there is formation of their button-like structure of the Hostoria, and hence it is called as the intercellular mycelium formation of their Hostoria or button-like structure. There is a very useful, very beneficial for the absorption of the nutrient inside that plant body. Then next thing is the direct with their aprosorium, or is a penetration pegs, means PP. Aprosorium means A. Intercellular mycelium means IM. Intercellular mycelium. First the spore there is a settle down on surface of this plant, and at the favorable condition, these spore they can be start their germination and first formation their aprosorium. There is formation of the aprosorium is a branch formation. After certain times, there is a penetrate the pegs, means they can be detached this surface of the leaves, and they start this mycelium inside that in epidermal zone, and after next time there is a formation of the germ tube continuously inside that cortex region, and there is a formation of intercellular mycelium, intercellular branching they can be developing to them. Then penetration through the natural opening. Stomata is there is a lenticell and hadathode. There is a natural opening through their any types of the spore, bacterial or virus or nematode, they can be penetrated to them. This opening of the fruit, stem or tuber field loose concentrated with the cell and allow their passage of air. Then the hadathode are permanently open spore at the margin at the tip of the leaves, and there is also very useful, very beneficial, and there is abundant moisture at that hadathode as well as lenticell as well as stomata, and it is so much favorable for entry of the pathogen, entry of the spore through this natural opening. This is the best picture penetration through the natural opening. First is these are the lenticell. Second thing is the stomata. This is the natural opening. Third thing is this is also in the bark and the lenticell. And fourth cell is a very well known about. There is a hadathode. Here the hadathode. Very well known about the hadathode. and there is a entry through this natural opening. Penetration through the wound, certain of this formation of wound, certain of this enzyme they secreted on surface of the leaves, and there is formation of the wound. This natural crack between this main lateral root, sort of the stem, sort of the branches, there is formation of wound that is also very useful, so much beneficial to them for entry of this pathogen for this now opening of the made by this fungus. Then infection after they can be germinate to them, then establishment of the pathogen with their vulnerable cell or tissue of the host and obtain the nutrient from them. The successful infection will produce the symptom after certain germination of the spore. Then incubation period, certain of the time they require for spreading of the disease. The time interval between the inoculation and the appearance of the disease symptoms, certain time, certain duration they can be required to them. Then latent infection, the state which holds the infected with their pathogen but does not show any symptom. Then growth and reproduction, the pathogen will grow and multiply within the infected host very fastly. These are fungus, they can be produced by the spore. Viruses, they are replicated by the cell. Nematode, they reproduce by the means of the eggs. They can be reproduced to them. Pathogens, dissemination of the pathogen. Pathogens are disseminated by the several way. First is by air. Means air is a major factor while airborne. This is a spore, they touch wet surface and get trapped, and air movement, they stop, and rain, they can be washed out from the one location to another location. This is the main factor by the air. And from the air, there is a brought down by a raindrop on surface of these leaves. There is brought down of rainfall. There is also a major factor. Wind also help the spreading by their blowing away rain. There is a flash droplet containing their pathogen also. Wind is also a major factor for spreading of this pathogen, spreading of this spore. Blowing from the one or blowing or fall on this rain of one raindrop, and inside a sporangia they can be detached, and they are easily, they can be flash out, they are drop out, and they continue huge amount of spore, they can be exposed to the atmosphere, and there is a easily dispersed from the one place to another place. Dissemination of the pathogen by water. Water is also a major factor. Pathogen in the soil, they can be disseminated by the rain, irrigation water that move on the surface through the soil. They can be moved to them from one field to another field. There is also major factor. At a rainy season, certain of the one field there is a disease of plant, and that disease, they can be flow of the water, the excess of water from the pass through the another field, or another certain of this next field, they can be passing through them. At that time, same time, these pathogen, they can be carried to them by this water, rainwater as well as the irrigated water from one field to the another field. Another, there is dissemination by the pathogen by the insect, nematode or other vector. There is also a major factor for transmitted by this insect during their feeding. Insect, there is a major primary food, there is a leaves. Also in the plant parts, this is nectary, they can feed into it. At that time, these disease, they can be carried to them from movement from the plant to another plant. Nematode can be also transmitted by the virus internally. Then dissemination by the pathogen by seed and transplanting process. From seed can be also transmitted to them. When they are diseased seed, they can be cultivated to them, and they can be packed to them, and that after certain time, certain of the seed, they can be disseminated from the one district to another district, one state to another state. At that time, these disease can be also transmitted through their seed. Transported to the other field, from one field to the another field. Then air sold and transport to the other area. Of the same area or another area, this seed can be also transmitted to them. Then by human also, human can be also disseminated or can be transmitted by this pathogen or this disease from one field to another field, one plant to another field, through the handling of the disease and healthy plant. Suppose in the human being, they can be handled to them, certain of the cut to them, certain can touch to them. At the same time, there is a disease plant, they can be touched to them, they are observing to them, and ultimately these so many spores, they can be fall down on the surface of the human body, and at the time of irrigation or at the time of certain of this working inside the field, at that time these spore can be disseminated or transmitted from one plant to the other plant, one field to the other field also. Tool, or there can be there is a pruning shear. At the time of pruning inside that tools, there are suppose there is a harrowing, certain of the tools they can be using to them, then pruning the certain tools they can be inside the field. Suppose certain of the human beings do not wash out, do not clean out by the tools, they can be using suppose one field, and these fields are the disease plant, disease spores, so many spores present inside soil, inside surface of this plant, and at the same the tools they can be using for another their field. At that time these spores can be also transmitted to them by using these different types of tool by the human being. Transporting by contaminated soil on their feet or their equipment, different types of the equipment, large equipment, harrowing, these are major equipment they can be used to them inside the field. At that time, when one field there is a infected to them, and these same of the instrument they can be using to them by harrowing for the another field. At that time, these spores can be also transmitted to them. Contaminated container, certain of these contaminated container they can be cultivated to them. They are contaminated already. There is a disease contaminated, and also they can healthy contaminated they can cultivate to them. These are also mixed to they can be carried to them by the disease from this contaminated container also. Using infected seed also. Nursery stock also, suppose you can seed are infected, then can you grow to inside the nursery, and these nursery plant can be distributed in so many fields, so many villages. At that time, same time these diseases also they can be passed through them. Then another is the epidemic. Disease epidemic in the plant can be in the cause huge loss of the yield. So many crop they tremendously are lost, tremendously decreasing the yield of the crop, as well as they are threatening to wipe out of their entire species, such as was of this case, and there is a Dutch elm disease. It's called a Dutch elm disease. It is an epidemic. There is tremendous loss, about 35 to 40% of the loss of the human being in each year. Throughout there, about $215 billion the loss in each and every year of the human being. An epidemic of the potato lead blight caused by the Phytophthora infection led to their great re-reach farmer and loss of the many their lives. Commonly, the element of this epidemic are referred as the disease triangle, and they are susceptible host, pathogen, and conduct to their environment also. For a disease occur, all three of these must be present. Below is an illustration of this point, where all these tree atom meet there is a disable or the disease. Fourth element missing from this illustration for an this is a epidemic of to occur in the time, specific the time they can be required to. As long as three of these elements are present, disease can be initiated, and epidemic will be only ensured if all there is a continue to the present. Any one of these three might be removed from the equation through. The host might out there grow susceptible there is a very useful, very epidemic can pass to them. As with the high temperature, idle their plant resistance, the environmental change, and not in the conducting for their pathogen to cause a disease or this pathogen. When the said tree or shrub gradually lost vigor and there is a display pale green or yellow color can be appear to them. Also small leaves they can be developing small bunchy leaves of the brinjal, grassy shoot of the sugar cane, poor growth also occurring to them, early leaf drop out, they can fall down the surface of soil, early fall, decline, color, and there is a dieback of the twig and branches. It is referred to as a tree decline. There are some of the decline caused by the specific disease and environmental traces, but there will be discuss of general decline which no specific cause has been identified. Over the spread of the air, tree or shrub and maybe subject to insect attack, disease, adverse weather condition, and other stressful environmental factor, soil compaction, change in the drainage, soil fill over the root system, mechanical damage to the root from the construction, and excess of salt accumulation are the best example of environmental trace factor affecting by the root system. Forecasting of this plant disease is spreading the occurrence of the disease in the epidemic or epiphytic form in a particular their area. Plant diseases vary in incidence from the season to season due to their difference in the nature and amount of the inoculum. Environmental condition, major factor, number and activity of the vector, also an other factor which affect the development and spread of the pathogen. If we recall in which the correlation between the result of the disease survival and the corresponding whether the condition in a particular area holds to be related to the biology and host plant of the pathogen. Forecasting of the plant disease occurrence involve all the activity in accelerating and is notifying the grower and community that conditions are there is a sufficiently favorable for certain of the disease that application of the disease the management measure will be the result in economic gain or that can be amount of the disease expected is an unlikely to be enough to adjustly or justify the expenditure of the time, energy and money and the disease management. Disease development is the result of the most pathogen environmental interaction. Therefore the enough must be known about the factor such as the nature, amount of the pathogen, inoculum, environmental condition that favor host there is a pathogen environmental interaction and leading to their successful disease development before the start and can be made at a forecasting and seasonal occurrence of this plant disease. Disease severity is also referred to as a disease intensity. For the diseases where the amount of disease varies greatly on different plant in the population, many of the arbitrary indicate the rating have been practical. They are usually discouraged and are replaced by percentage scale and standard area their diagram of the disease intensity. Then remote sensing. Remote sensing is also a major factor. Sensing of provided by powerful tool for detection and measurement of the disease. How much disease can be spread to them? They can be using to this remote sensing. It is used to survey a large crop area by using this aerial photography. Disease, disease plant can be identified as a distinct their patches in a otherwise uniform their picture. It is useful for measuring a disease such as a late blight of the potato where early disease foci can be also located and there is a subsequently spread is follow. The remote sensing is also useful when the sudden disaster strike and rapid there is a appraisal of the situation is required. It is wheat crop aerial this is a photography can be used to determine the extent damage take all or eye spot disease. Similarly, the incidence of the heterobasidium can be also monitored and find relation covering and is undulating their boggy or rocky land. Student assignment, write the information of the penetration, define the infection. In the next lecture, we will discuss about the introduction to this plant pathology. What is meant by host, symptom, parasite, pathogen, inoculum. In this lecture, we will discuss to them about this what is my host, how to symptom, different type of symptom, post they can be appearing to them, and after they can the parasite of the disease they can be occurring on surface of this plant. Then pathogen they can be causes the disease, and inoculation there is a different inoculate to them. Mostly in a natural inoculation means there is a force they can be surface of the natural opening like stomata, lenticule and hydrozoa they can be inoculate to them and they can be these spore they can be sprayed to inside that mycelium or this Hostria formation. Then in the next lecture we will discuss about the defense mechanism. Naturally plants can be developing by the defense mechanism. They can be survive to them. They can be tolerate to them by this defense mechanism for any types of the disease. Then type of the defense mechanism. First is the naturally certain of this plant they is healthy, they tolerate to them, they can be do not grow properly of the disease. Only certain of the part they can be grow to them, but no any side effect on this part, other part of this plant. Another is the certain of this plant part, they release certain of this chemical substances on surface of this plant and hence these spores are do not germinate to them. In that type there is a different mechanism we will discuss in the next lecture. Thank you so much.